Hi everyone, welcome to this video which I'm super excited to do because today I'm going to wrap up the short trial that I did looking at answering the question, does soil microbiology work? Now this is the third part in a mini-series I started a few weeks back, so today is going to be the concluding episode where I actually answer the question, does soil microbiology actually do anything for plants or not? Can we prove it and show that this is the case? Or is it a myth? Well, we're going to get stuck straight into this question now with the final set of information relating to that trial, and that was the soil tests that I had kind of conducted by my laboratory at the end of the grow period. But before we do that, just to recap, what I did was took some soil from kind of a typical agricultural setting. I treated some of that soil in order to reduce, I actually wanted to sterilize the microbiology, but I reduced the soil life in one of the sets of pots to minimize the amount of microbes there, and I let the other kind of grow naturally. I then put some cress seeds in them and allowed them to grow before harvesting the cress and doing some analytics. So those analytics was weighing the kind of cress, looking at the amount of organic matter that was produced, and also looking at the bricks as well. And again, just to recap on that, the naturally grown, so the cress that was grown in the more biologically active soil had quite a bit more substance to it, more mass. So it seemed that the soil life was in fact helping the cress to grow better than the more sterile soil with much lower numbers of bacteria and fungi. When it came to the bricks, however, it was a slightly different story, and there was this nagging question where actually the more naturally grown cress seemed to have a slightly lower bricks level than the more sterile soil. And actually, anecdotally, I was talking to the guys in the laboratory this week, and one of the scientists in fact told me that the cress which I'd harvested and put in the fridge, the sterile or lower microbiology cress plants had lasted much longer in the fridge storage than those that have been grown naturally, which is interesting. I guess it relates to the more sterile nature. There were less organisms present on those particular stems of cress, so they weren't organisms there to start kind of that breakdown process and turning the plants into the kind of mush that you would expect for something to perish after time. So a little bit of difference there, but there is this question about the bricks still, and maybe we'll address that in a little bit. But for now, let us have a look at the soil results, and I will talk you through what I've seen and perhaps what this means. And so here are the results. It's pretty clear looking at the numbers that there's quite a wide range of difference between the natural samples and the more sterile or low biology samples. I've highlighted here in green, that's obviously the maximum for each kind of metric. The red is the minimum. So going through one by one on the moisture content, overall there's quite a large range. It goes from 33% moisture content up to 38%. The samples really were quite wet at the time of analysis. The guys did even comment on that in the laboratory, how damp they were. So that's kind of a range of 5% difference for the natural from the lowest to the highest, whereas the sterile is actually a little bit more, a slightly higher variation, just shy of 29% to 35%, so 6% variation, so a percentage higher in the sterile than the natural. And actually I would need to check, but I do seem to recall that there were some similar kind of levels of variation in terms of the weights of the cress between the two samples as well. Next up, looking at the active bacteria. In fact, we'll talk about the total bacteria, which is the next one along first. So just to remind you, this is the total reserves of bacteria in each sample. So that's the active and inactive bacterial organisms. Again, there's quite a large variation, 292 up to 420. We'll kind of call that 300 to 420. So there's 120 micrograms per gram of soil difference with the natural samples. On the more sterile samples, there's a lot lower variation, but as you can see, the numbers themselves are quite a bit lower anyway. So we're going from 190 to 250, so around a 60 microgram per gram difference there. So the active bacteria, just before that I skipped, this relates to the alive, functioning, active bacteria. So they represent a part of the total bacterial population, but it is just the bit that is working away at the time of measurement. In the natural samples, quite a tight band of stats for those. So we go from 44.8, 45. Actually, I skipped one. We go from 44% to 45.5. So there's around a 1.5 microgram per gram difference, which is really quite surprising. It's as similar as that. The sterile samples go from 3.3 to just shy of 8. So there's kind of around a 5 microgram per gram difference there. So slightly wider variation in those samples. We'll just do the same thing quickly with the total fungi. I won't describe what that is. It should be obvious if you kind of understood what I said about total bacteria. The ranges there go from 330 to 470. So again, another 140, 150 microgram per gram difference with the total population there. And the active ranges are around 21 to 65. We'll call it 20 to 65. So 45 micrograms per gram difference. So a little bit wider difference in terms of the smallest to the largest for the natural samples there on the active fungi. Whereas on the sterile samples, a very tight band for the total fungi, 270 to around 300, so 30 micrograms per gram difference. That's really quite narrow. And similar story on the active fungi as well, 2.1 to 2.31, so not even a full microgram, around 0.2 difference. So there's some quite interesting variations, some of them slightly bigger, some of them really quite narrow. So what can we say about these numbers in relation to these samples? Well, actually, I think that where I've highlighted kind of in green and red tell a bit of a story. There's a slight anomaly perhaps in terms of sample natural 3, which has quite a bit higher proportion of bacteria than the other two samples, whereas the sterile kind of trio is a bit narrower banding overall in terms of the microbiology. So I guess where the populations were initially kind of nuked in the autoclave and set back at the beginning of the trial, it kind of appears that things have developed fairly evenly and in proportion over the duration of the growth of the cress across the 3 samples. So pretty even, and actually the worst sample or the worst characteristics were all within one of those samples, and wasn't quite the same case with the natural samples. I suppose perhaps we would have liked to have seen all of the best characteristics within one sample, but we kind of got 3 out of 4 measures there as well. So yeah, in kind of close of this, I think the more natural samples, very interesting, and they're really actually not too bad values in terms of where you might want to get samples to start delivering real kind of benefits to what you're growing. But the sterile samples, I think the key takeaway with these is that where the plant is the driver in the soil, and I've talked about this before, the plants will actually give out biochemicals, foods, things called exudates, which encourage and stimulate particular groups of organisms. And it seems that this has happened in a fairly even and consistent manner, which has allowed sterile 1, 2 and 3 to kind of evolve their microbial populations at a roughly similar rate. Moisture content is incredibly important because there's quite a narrow band. You don't want samples too wet, you don't want them too dry, and these organisms, like I've talked about before, need moisture in order to survive, to eat things and to reproduce. So it often goes that the most moist sample under certain conditions is the most active. You can see here with, the green highlights, that's not actually necessarily the case here. But that said, they're all in a very similar band, the sort of upper 20s to upper 30s range. So I perhaps wouldn't expect quite so big differences anyway. So just by way of quick summary on the soil microbiology testing, these are the average numbers. Again, you can see actually the natural soils had a little bit more by way of moisture content, but in my experience from looking at other soil tests, that would not explain such a wide differences in terms of the values that we can see for total inactive bacteria and total inactive fungi. That is purely down to the preparation of the soil at the start, so the killing off of organisms and the time under which they were growing to be stimulated by the plants that were in them. So as I've just said, I'm pretty happy in terms of the natural sample values. These are pretty good actually. They're not in a bad range at all for such a short-lived, fast-growing crop that's going to be harvested really quite quickly. So, in close to these, I think that when we consider the amount of organic matter that was produced, the soil figures mimic or enforce that really quite beautifully. Now, in terms of the difference in the Brix values, which I mentioned in video 2, there's a whole host of reasons why this could be the case. I personally think the two most likely candidates are the additional root growth that was present in the more natural samples. I actually think in retrospect, if I had done this trial again, I would probably wash away most of the soil. I would take my soil sample and then wash the remainder of the soil away and measure the complete mass of all the plants that were left, so not cutting them to soil level at all. I'd measure the whole thing and then I would compare the sterile with the natural on a like-for-like basis. So the natural samples, the plants actually had really quite a substantial amount of additional growth beneath the soil and actually coming out of the pots as well. So in terms of the reserves, when we consider the sap or the percentage suspended solids, there's actually a bigger body there for the plant to try and maintain. So that would probably explain why the percentage suspended solids, the sugars, would be a little bit lower than the more sterile. I mean, there was only 0.5% in it, but that could be one explanation. That said, I've done some other videos recently looking at the Brix values of different foods. These levels, 1.5% and 2%, are very low indeed, and I think that's reflective of the timeframes within which these plants were grown as well. So we're looking at little over 7 days. The other factor which I think could form part of this as well, and I've talked about the relationship between plants and the organisms. plants will give out biochemicals, exudates, which are simple sugars, to feed and stimulate the organisms in the area around the roots. And the microbial populations, as you've just seen in the soil data, were really quite substantially larger than the sterile samples. And so there's a lot more mouths to feed, and therefore there's less available for the plant at this particular point in time. But it is a very short space of time. If cress was a kind of much longer living plant, if we were to keep them alive for a number of months perhaps and remeasured, we might find different results again. So I think they're the two most likely ideas as to why there might have been a subtle difference, perhaps not how we would have liked within the bricks values, but I think now we've had the soil data, it kind of shows a little bit more of the background behind that and possibly gives an explanation as to why. But what do you think? I would love to know your thoughts. If you do have a theory, do put it in the comments below. I will read it, I will get back to you, and perhaps others might have comments to add as well. So yeah, let me know your thoughts. So there you go, there are the results. I think all in all, I couldn't have planned this any better, and there's always a risk when you're trying to demonstrate a point that things can go off at a tangent and they don't necessarily prove the principles or the things that you think they will. And certainly when you're working with nature, natural processes, this can often happen. Bacteria and fungi, a little bit like don't work with children and animals, things can go wrong. It's the same with bacteria and fungi. They can do some weird and wonderful things, being governed kind of more by the random laws of nature than anything. But overall, I'm really, really pleased with how this turned out. It illustrates the point perfectly that yes, soil microbiology does help plants grow. And so you can take this proof and go and look at how you might apply it. And I've mentioned in the other videos, perhaps you can do your own trials to kind of look at the effects of with and without in your vegetable patch and see the effects that it can have on the food that you're growing. And as I keep saying in my videos, it's this abundance of healthy, diverse, kind of bountiful, active microbes that are the key to unlocking the potential of your soil and bringing those nutrients that your plants need and giving them the resilience they need to be the healthiest they can be so that you can grow the healthiest and tastiest food that you can. So I really do hope that you've enjoyed this video. I've really enjoyed doing it, and I think I'll probably do another test or trial in the near future, either to replicate this in a slightly different way or pick up on some other ideas and principles and demonstrate them in a very tangible, real world way. If you have any ideas for trials or have something in particular that you would like testing, drop me a comment below or send me an email. There's a link in the bio. I would love to hear from you. But for now, that wraps this video up. Hope you enjoyed it. If you did, please give it a thumbs up. If you found it useful and want to see more things like this, do consider subscribing to the channel, where I put out lots of videos each week based on soil microbiology and using it to get the most out of your soil for healthier, tastier food. But let's wrap things up, and I look forward to seeing you in the next video. Bye.